I decided a couple of things. So first of all, last night under deep reflection, I changed the lab schedule again to after I've heard the decisions of folks. I changed it again to represent what we had decided to do. Um, I don't know where Devin is, but so I've sent you all that. They're also posted on Blackboard, so you guys can take a look at that. Mostly, I put in color. I put in color what has changed, right? So what I decided to do was because today in lab we're going to do two biochemical tests: the nitrate reduction test and the urea hydrolysis test. And then that's going to be the end of the material for the second practical. So because we decided to have the practical after spring break, what I did was I moved some Unit 3 stuff up and I broke it apart so that next week we would start Unit 3. But then when we come back, we'll have the first Tuesday we come back from spring break so that we'll review for practical. Okay. And then after that, we'll take the practical on Thursday. If you can't get in there, mommy, if you can't get in there, then just go to announcements, or you can go to Blackboard and look at the schedules or the lab and go to lab schedule, and that's posted there also, so you'll be able to get those also. Okay. So the things that we do next week are part of. Yeah, you have access because I gave everybody access. It might be your individual computer, but okay. Well, then go to schedules or go to labs, and they'll be there also. Okay. Yeah, I don't know. Some people's, I don't know. Blackboard's kind of funny on some computers. I have no problem opening it from any computer in the house. But anyway, so so the lab schedule will be augmented a bit, um, and then that'll give you guys an opportunity to have a restful spring break and not really worry about things over spring break unless you want to. I might go ahead and post the take-home exam for exam 3 before spring break, and in case anybody wants to work on it over spring break, but that'll be at your own leisure. Okay. I also sent you a video from one of my favorite mycologists. His name is Paul Stamets, and he's got some really great ideas. So if you get an opportunity, you ought to take a look at that video, because the content of that video will be on the exam. The content on that video really deals with the Presidio Mycoda, which are the mushrooms, which we're not going to address too much in this course because, yeah, sure. But we're not going to address too much in this course because they don't have a lot of clinical relevance. Some of them do produce toxins, so we'll address that. But for the most part, we're going to be looking at fungi that can cause infection. Okay. All right. All right. So let's get started then. So if we look at the fungi, oh, by the way, I decided not to talk about the bacteria yet. If I look at the schedule when we go to spring break and decide I can put it back in, I will. But we're going to cover the bacteria in such detail when we get to infections later in the semester that this would have been an introduction to them that way, but you guys know a lot about them already. So I decided to mostly look at the eukaryotic organisms and the viruses, right? Those things that are not alive in this particular unit. And that way we will pretty much catch up where we need to be in order to finish the course on time. Okay? All right, let's get going. So if we think about the fungi as a whole, right? The study of the fungi is called mycology, right? And myco stems from the fact that these particular organisms, most of them will produce filamentous somatic structures that we call mycelia. Okay, and we'll talk about what that is in a minute. Okay, but so they belong to the kingdom fungi. They're chemoheterotrophs, and so what that means is they have to digest and use nutrient sources that are mostly organic. That's right, mycelium. Okay, the organisms in the fungi, there are three groups, right? They're the yeast, which most people don't know that yeast are fungi, but they are. They're single-celled. And then there are the molds, which are multicellular and have mycelia. And then we have the Presidio Mycoda, the macroscopic organisms, the mushrooms, the toadstools, the puffballs, those things. And they all have mycelia, right? And so you can see that there are three different groups, and the three different groups are very unique. And so this is my favorite kingdom of the eukaryotes. I like them even better than I like animals, and I love animals, but man, this is my favorite kingdom. They're just so cool. They do so much for the world, right? So therefore, if we think about them, then there's a variety of different arrangements by which their cells show up as either single-celled, unicellular, or they show up as tissues, right? Filamentous somatic structures are fleshy, right? So if you think about that, there's all these different types of organisms out there that belong to the fungal kingdom. And we're about to get to the time of the year where they show up in great numbers in nature. So the early part of spring, they go crazy. And then in the fall, they go crazy. They just start growing all over the place. So I'm thinking that I might get some stinkhorns. And so if I do, I'll take some pictures of them, and then I will show you what stinkhorns are. Okay. So they are all absorptive. So they don't have mouths. So they have to release exoenzymes, much like the bacteria would, and then they break down organic substances around them, and then they bring in those smaller molecules into their cells to which they can use them to undergo cellular respiration, right? The really cool thing about most of these organisms is they have a sexual and an asexual state. Now, some of them are only asexual, like the bacteria, right? But a lot of them can have a form of sex where there is an exchange of genetic information, and therefore the spores that they produce, the sexual spores that they produce, the diploid sexual spores that they produce are unique genetically, right? But they do not form embryos. Okay. Some of the characteristics about the fungi is that they are the number one natural decomposers of the world. They are the recyclers of nutrients in the world. So they take anything that is organic, that's in nature, and they break those things down. And not only that, but that leads to nutrients being widely available for all other organisms in nature. So they help to facilitate the transfer between a whole bunch of different organisms within nature, right? So it's not just like they're themselves, but they're, to me, the fungi basically, they don't think, but if they were thinking, to me, they would think like this: I'm gonna feed myself, but you know what? I'm gonna feed everybody else in this world, because that's what they do. They mitigate nutrients in the forest and in nature. They really do a good job of that. They are not vascular. Uh-huh. They they love they can take in nutrients, but they are not vascular. They don't have xylem and phloem, right? So they are not vascular structures. But each of the cells work individually, and we'll talk about those in a minute, right? Now, the fungi themselves, in lots of different formats, and as both yeast, as as yeast, as molds, and as macroscopic organisms, are used for food. They're used to create food. They're used to create flavor, and they're used in religious ceremonies also. So there are a lot of these things that some of our First Nations people will actually consume. That's right. They'll actually consume, and they get into a higher state. They get high, if you will, and then they believe that they can come in contact with their ancestors and get direction for how the tribe should be doing things based on, well, okay, but those are different, right? Peyote is not really a fungus, but yeah, but there are some magic mushrooms, if you will, that will allow people to get into a higher state. Um, yeah, and I also have some friends in Colorado and in parts of Arizona, Devon, and New Mexico, around the Cruces area, where their particular tribes also use this, right? So I've in my PhD program, I had a couple of First Nations folks that are from those areas, and they talked about using these particular things as ways to get them. Oh, I see what you're saying. Yeah. Okay. Um, Anyway, the first antibiotics against bacteria were produced by fungi, and so therefore they have the names of the fungi in them, right? So the mold Penicillium produces the antibiotic penicillin. The mold um, Cephalosporus produces the antibiotic cephalosporin, and there's a whole bunch of these out there, right? So we took a lot of the first antibiotics that we utilize and learned from the fungi, from the molds, um, how to use these things to fight bacteria. And now, of course, we can make a lot of the antibiotics and by molecular techniques, right, in labs, so we don't have to rely on them. Although one of the last opportunities I had to learn and be part of an organization was I was part of a little outfit called AOK Abayo. They are a vaccine company, and they're found right here in Pflugerville. And I worked for them for about 3 years, and we grew our own fungi and we extracted our own antigens from those particular fungi. So it's a pretty cool operation. The fungi are really important research tools because they're eukaryotic, but they grow more rapidly, they mature more rapidly than animals do. So using their cells to look at pharmacokinetics, pharmacodynamics, and a bunch of different kinds of things about how cells grow and things like that. They are really important research tool. About 30% of all of the fungi can produce disease um, in plants, animals, and humans, but mostly in plants. Uh, so there are only 5 true fungal pathogens, right? Um, So we'll be talking about 4 of those because one of them is only found in South America, and so there's no reason to talk about them unless you're going to go down and live in South America, right? Because they prefer acidic environments, the fungi can spoil fruits, pickles, jams, jellies, juices, coffee. They can grow in just about every environment that's acidic where bacteria just simply will not grow. What you're looking here at this image is a nematode catcher. So this is a mold that is in the environment, and it has these little hyphal loops, right? These mycelial loops by which it can capture the nematode, right? And it will digest that nematode alive, right? And so it's a pretty cool thing to think about how that might happen. Some of my important mushrooms that I like to go hunting for are morels. So this is a picture of them right here. Right. They're delicious. One year, one year we found so many we had to cook them right there because they were so beautiful. I won't pick them all, right? So they do mostly on the eastern part of Texas, but and they're kind of small, Devin, where other places in the United States and other places in the world they get fairly large. But uh, oh yeah. Oh, don't, but if yeah, I promise, Devin, if you show me, I won't take them all. I'll only take a few, because just like with any fungus, I will allow for the fungi to mature so that they sporulate. And then when they mature and sporulate, I will take the fungus up out of the ground, and I will help to spread the spores all over the environment so that they grow. One of the benefits of buying the property in Bastrop is I have a lot of fungal species in that forest, right? And so, oh, yeah, yeah. So if you do not know what you're doing, mommy, do not go pick mushrooms for dinner, because yeah, the chances are most of them um, are going to be poisonous, right? So some of the fungi will produce haustoria, and that's a really great word. And I call these nature's natural vampires, right? So if this is a cell, right, it's a eukaryotic cell. The the fungi have these mycelia, and the mycelia can get into the cell, and they can just take everything out of the cell that they can use as a nutrient source, and therefore, um, to me, they're nature's natural vampires, right? Yeah. So Caitlin, I grew up in Corpus Christi, Texas, and so when I was growing up, there were only a couple of things you could do for fun as a kid, right? And that was, you could go to the beach and surf, or you could go out to the cow patties and pick the shrooms. You could boil the shrooms up. You could make a soup out of them, and then you could air surf, because you didn't really need any waves if you were on shrooms, right? You could just feel like you were surfing. And many of my friends, unfortunately, had some pretty bad reactions to the shrooms. So for those of you guys who are thinking about using shrooms, I would advise against it. There's a very small therapeutic window between when the shrooms can be euphoric to where they become toxic to the body. And so you just have to be really, really careful. And by the way, if you get caught and you plan on going into a professional program, you will be blackballed and you won't be able to get in. So be careful. Okay. So almost all the fungi are aerobic. They need oxygen, right, with the exception of the yeast. So the yeast are facultatively anaerobic, and therefore they can grow in an anaerobic environment, and we use them in that process um, to make beers and wines and all kinds of different things like that. This is a bird's nest fungus. Ever seen this, Devin? This is one of the coolest fungi you've ever seen. And they're little tiny things, right? So they're probably a little bit smaller than a penny, and most people would never see them. So last spring, I was walking in the park, and I looked down, and the bird's nest fungi were everywhere. They were everywhere, and most people didn't see them because they're so tiny. And if you're not really attuned to checking out nature for microorganisms like I do, you remember that? You remember that movie that what was it, The Sixth Sense, where the little boy said, "I see dead people. I see fungi everywhere. I see them everywhere, right?" And because I'm so attuned to looking for them, I see them everywhere. But they were everywhere, and it was only there for about 2 weeks. And so if um, if they show up again this spring, I will take a picture of them. And then Devin, you kind of know where I live, so you can go to the park if you want, and you can check them out yourselves, because they are just phenomenal little creatures. Um, that um, yeah. Okay. Questions. All right. So if we think about them, there are three groups to the kingdom fungi. Now, this is an older spelling of the kingdom, right? Nowadays, it's just this, right? So as people get more accustomed to using different terms, this is an older way to do it. It's still okay, but this is kind of the newer way. So it's the kingdom of fungi. Okay, there are three groups: yeast, which are single-celled, right? Molds, which are multicellular, and they have somatic, filamentous somatic structures, right? So this is a plate that one semester when I was lecturing on the fungi, the students asked, well, what is this stuff that's in the air that makes us sneeze all the time? I said, well, there's one particular organism that we typically talk about that's a mold, and that's Altenaria, right? It's one of the most common molds in the air, and it makes you sneeze, right? And so there it is right there. But I told them, I said, but there are thousands of species of mold spores in the air, and they're just they're there all the time. I said, especially right after a rainstorm, they go crazy. And so they didn't believe me, so I took this plate and I stuck it outside. Have you ever been to Riverside? The science area is in Building A, and up there by where I live, by the 1406 of the micro lab, there's this little walkway that you can go out the door, and it's up on. You can kind of walk down the stairs and leave the building. So I took a plate and I stuck it out there for about 20 minutes while I was lecturing. Then I went back, and one of the students, I said, just cover that plate, and then we're gonna put a piece of tape on it so that the cover doesn't come off, and then you can sign it, so I didn't get in there and mess with it. And so we put the tape on, and then she signed it. And then we came back the next week, and she said, "Can we look at the fungi?" I said, "It's only been about 4 days. Let's give it another day or two." And so Thursday, we went into the incubator and pulled it out. And ladies and gentlemen, there were over 200 different species of molds growing on this plate. It is it's an outstanding representation of the amount of mold spores that are in the air. Okay, so they're there all the time, right? Here is a stinkhorn. So a stinkhorn is a mushroom that emits an odor like poo. And so flies and beetles think lunch. And so they come on and they jump on that particular mushroom, stinkhorn, because they think it's poo. They think it's lunch. But when they figure out there's no lunch there, the mushroom um, has had the insects, the flies and the beetles, do exactly what it wanted them to do, is to crawl around their lamellae. These are the lamellae, and pick up spores. And so when they pick up the spores, they fly away or they crawl away, and then they take the spores and they drop those spores off in different locations. And so that's one of the ways, that's one of the mechanical means by which the spores of these fungi get disseminated into the environment. It's a beautiful piece of biology. Any questions about the three groups? The yeast, right? The molds and the macroscopic organisms. We're mostly going to be looking at this group here because they're the ones that cause the most troubles to humans. This one has a few pathogens that will cause problems, but these guys, they almost never cause problems for humans unless you take them and they are toxic. Then there can be a problem, and we'll talk a little bit about that, but not so much. Okay. So if we think about the yeast themselves, they undergo mitosis. They bud laterally. So one of the key indicators that it's a yeast is if you see budding. And so here's a parent cell right here, and there's a bud, right? But here is, you can see this particular yeast cell right here is budding on two different positions, right? These are bud scars, and you can see bud scars. And some of the older yeast, if you look at them real closely, they'll have lots of bud scars on them because these cells, as they're living, they will produce a bud about every 45 minutes, right? And so eventually what happens is they do die off, but by the time they die off, they produce so much progeny that that they that they have continued to survive. This thing here is what we call Pseudohyphy. So these are yeast cells, and they're producing these elongated structures, but this is really an elongated yeast. We call it, it's termed Pseudohyphy. That's right, Nayeli. Pseudohyphy is found in a few species of yeast. The most prominent and most important one is Candida. Or if you're from the north, Candida is what they call it up there. But Candida is a common yeast found everywhere, and there's been a lot of articles that have been written about Candida that basically say people can become colonized with it, and that is true. But most of the time, it's just people's misunderstanding of what that means. Because in order for these organisms to colonize the body, there has to be some kind of disruption to the normal inhabitants of the GI tract. And so let me give you an example of that. One of my best buddies in high school was a debutante. So she had beautiful skin, and the reason she had beautiful skin is for most of her life she was taking tetracycline. So she could never go to the beach with us because she would burn really badly, because if you take tetracycline, you become very sensitive to the sun. But anyway, so she took tetracycline for most of her life as a young adult, so that she wouldn't have any blemishes, because she had to go to a lot of parties and wear a lot of evening gowns and deb gowns and things like that. And she couldn't have any of these blemishes on her body. But that had a terrible effect on her as an adult, because her esophagus, um, yeah, it is. Her esophagus is completely colonized with Candida. And what that means is that there is a biofilm in her esophagus of Candida. And so it does interfere with her ability to do certain things. So every day now she has to take an antifungal. So she wakes up in the morning and she takes nystatin every morning to combat her biofilm, so that her biofilm doesn't go crazy. Because it we don't we the I don't, but I'm not her physician, but her physicians don't want the yeast to make it into her intestinal tract and then cause a biofilm on her intestinal tract. And her intestinal tract is still very, very diverse, but her esophagus is not. And so she, from time to time, has problems with the inflammation and the effects of having Candida overgrowth biofilm in her esophagus, right? And that's just from simply causing a disruption. Okay? Questions? All right. So these are the molds, right? And the molds are and the larger organ, the macroscopic, all have mycelia. Now mycelia shows up in two different formats. It's filamentous, and it shows up looking like this. It's multinucleated, right? And so these are the less evolved molds. And you can see that this one, compared to this structure, which has cross walls and then is multinucleated, those cross walls really kind of divide up the different cells within the mycelia. And one of these segments is called a hyphy or a hypha. Okay. So a lot of people make the mistake of basically saying that moles have hyphae. And what they really should say is that moles have mycelia, right? And then one particular segment within the mycelia is a hypha. So these two different presentations of the mycelia, right? This the filamentous somatic structure, that real kind of fluffy stuff that's growing on auger, or on if you all of a sudden decide you want an orange, and you go to the crisper of your refrigerator, open it up, and then your oranges are covered with green mold, right? Yeah. So I've never seen it said that, Hyphy. Yeah, but I've never seen it said that. But these different presentations, this particular type of mycelia is called aseptate. It's also called, oops, darn it, aseptate. That means it doesn't have cross walls. This one is called septate. And the septate organisms are more, if you will, they're more evolved. They're higher functioning than these guys are. Okay, so we'll talk about the differences. Another term for aseptate is cenocytic. Man, those are hard to see on this gray. Cenocytic. They mean the same thing. They mean no cross walls. Okay? Questions? All right. So the molds and the macroscopic organisms grow radially. What does that mean, Devin? If I was to put a little bit of fungal elements on media, they grow outwards like this. Are you with me? So all of the patterns of the molds and the macroscopic organisms are gonna look like this. And so the mycelia is deep in the ground, and so you have these things growing. But then eventually what happens is you have the mushrooms pop up, and the mushrooms are the reproductive structures of the mycelia, right? So when you see the mushrooms growing, it is the reproductive nature of the mycelia itself. On our body, it would grow like that also, right? So if they're growing on our skin, they would grow radially, right? And so, in kind of a weird pattern. But then what would happen is you would see that pattern kind of growing where it was growing, and we call that pattern ringworm if it's growing on our things. Okay? So, but they do have that pattern. Here are some of the macroscopic organisms, right? So the mushrooms, the toadstools, the bracket fungi, the stinkhorns. Some of them are so beautiful and so dainty and so fragile that if you just blow on them, right, they break apart. Some of them are so rugged that you'd have a hard time pulling these guys out of this particular pole, right? That's right. That's right. So chicken of the forest or chicken of the woods, it grows like this, right? But you just got to know what you're looking for, right? So this particular structure here um, is the reproductive structure of the macroscopic organism. The mycelia is throughout the entire tree or whatever, right? And so therefore, these are showing that that tree is weakened, right? Yeah. So you have to be careful. And some of the bracket fungi, like for instance, and the place that my wife and I used to live prior to where we live now, we had a beautiful oak tree, and that oak tree during a really bad thunderstorm, um, we lost probably, about 10% of the tree because one of the big branches broke off. And so I I went ahead and trimmed the tree and cut it back and treated it. So I painted the wounds. And by the way, A&M just published a paper that said that if you have oak trees and you're trimming them, or they have wounds, you need to paint the wounds immediately. So before they were saying only during kind of the more temperate parts of our calendar year, you needed to paint the wounds because the beetles will get in there and they'll put oak wilt in there, the fungi that causes oak wilt, and then you'll have oak wilt. But now A&M is saying because of the changes in temperature, I can send you the article if you want. But what they're saying with the changes in temperature, that no matter what time of year, we need to paint the wounds of the oak trees so that they don't develop oak wilt. Because if you have oak wilt, it will destroy the tree very, very quickly, right? But also it spreads through the roots. And so you know how the roots of trees are as big as the canopy. And so therefore it's going to spread throughout the environment and you're going to have a huge problem, right? The fungi themselves are not trying to cause problems for us, but they're just trying to mitigate and make nutrients out of things that are weakened. And so, you know, they take one tree and they turn it into nutrients, but in the process of doing that, sometimes they can take out a whole forest. And so you have to be careful. So here's an example. Yeah, they're just doing their thing. That's right. So here's an example of a fairy ring. How many people have ever heard of a fairy ring? All right. How many people have ever heard of a fairy ring? So the fairy rings are really about, you know, folklore basically says that the fairies were running around and dancing and dropping off their fairy dust, right? Tinkerbell. And then the mushrooms would grow where they drop the fairy dust, right? Which is a fun thing to think about, but that's not at all how it happens. Ladies and gentlemen, the center of this colony is right here, and the mycelia is throughout the ground so that it's radially growing all over the place. I think you get the hint by now. And so the mushrooms that are popping up there are the vegetative structures. I'm sorry, are the reproductive structures of the mycelia themselves. Yeah, Julie, they can be miles long. They're huge thing. So I did send you one of my favorite videos of Paul Stamets. It's called 6 Ways That Mushrooms Can Save the World. That is learning material, right? So I want you guys to take a look at that because that stuff will be on the exami poo, right? And we can talk about it as you learn about it. But also, I'm in the process of developing your next take-home exam, and thinking about what I was going to do for the rest of the course. I decided to really, I was going to do parasitology at first, but I think I'm going to do mycology as kind of your take home. And so I'm going to come up with a case scenario for the fungi, and you guys are going to be doing this as your take home. Okay. Any questions? Any questions about how the fungi grow, their
Is it
mycelia, their reproductive structures, why they're important? Yeah, go ahead, Calvin.
is it true that the mushrooms that we see, they're like the fruit of the plant or whatever, and then the so it's kind of like an apple on the end of a branch of a tree?
Yeah,
Is that
well, so the mushrooms are the fruiting bodies, right? For sure. But they're not the true fruit. The true fruit are the spores. If you want to make, if you want to make, if you kind of want to make kind of a comparison to what that is, because the spores are what will produce progeny. When we talk about fruit, we talk about things that produce seed, right? And the seeds are what the fruiting bodies are really about. The fruits themselves are just nutrients for the seeds, but in this case, you're looking at the spores of the mushrooms to be really the true seeds, if you will, if you make a comparison. Okay,
thank
Calvin?
I have a question too. I'm sorry, I'm still having a hard time understanding the relationship between the uh, mycelia and the hyphae. Can you go back a
Okay.
couple slides when you had the two next to each other?
Yep.
I don't really understand. Yeah, this one. So,
Okay. Well, you can't. I'm gonna have Julia. I'm gonna have another slide coming up in a little bit that's gonna really show it microscopically, so you'll be able to see them.
okay,
Okay.
okay, then I can wait.
So
Yeah.
here the folks are just calling it hyphy, right? But that's not really correct. I mean, I guess it's not incorrect, but it's not really the way we, as mycologists, I'm a trained mycologist, so it's not really the way we talk about them. Hyphae are one single section in a mycelium, right? So if you
So
think
is it like
about,
a mycelium is like a chain of hyphae, kind of?
that's correct.
Okay.
That's exactly right. So
Okay.
if we look at this section of mycelia, right, then each of the nucleated sections are hyphae.
Okay.
Okay?
Gotcha.
All
Thank
right.
you.
All right. So this is ringworm. So guess what? The center of that fungal infection is right there. And guess what's growing? Guess what's growing right underneath the skin, right? Of mycelia. Hmm. And so if you look at this, it's real scaly kind of in the middle, but on the end, there's these little pustules, right? These little pustules all over. And that's where the fungus is actively growing, right? So it's growing radially. It's going to continue to grow that way. And so it has this irregular ring, and that's why it's called ringworm. So, some people think that ringworm is a worm. No, it's not a worm. It's a fungus, right? And we're going to talk about them a little bit later. Okay, questions? All right. So, oh my goodness. So, one of the, Devin, what's this, Devin? Yeah, no, but yeah, you just gave. So these are some of the most beautiful edible fungi that there is, right? So yeah, so this is Wiltacoche, right? Wiltacoche is corn smut. That's right, ladies and gentlemen. It is corn smut, and it is some of the most delicious fungus you can have. As a matter of fact, if you find your corn with smut on it like this, these go for about $150 a pound, right? Where corn goes for, I don't know what, it goes for what, 30 cents a pound or something like that, right? Yeah. What is smut? Smut is a type of a mold. Well, actually, it's a type of a macroscopic organism. It's a fungus, right? So here are truffles, right? And so you can see, I had a friend who was growing mushrooms in Wimberley. I don't think he's growing them anymore. He had troubles, but he had a couple of hogs. And here's this picture is showing dogs. I didn't have a picture of his hog. His hog was huge. So he had truffles growing in his ground, and every so often he would call me out, and we'd go truffle hunting. And so we could go in, and he somehow or another got the truffles to grow. I don't know how he did it, but he did. So, um, but he would have his hog, and his hog would smell the truffle. They have really good sense of smell, and they'd start digging, and then we would try to get the hog out of the way. And this thing was big, and he wouldn't move all that much. But if not, then that hog would eat the truffles, and the truffles themselves sell for about $300 a pound, right? So there are white truffles and black truffles, right? So when you think about, most people, when they think about truffles, they think about chocolate candy. That's just a marketing ploy, right? Truffles are fungi, and they are delicious, but they're so expensive. When my wife and I were in Vegas one year, we were in the Venetian, and we were running around, and I saw this restaurant, and it was basically touting how good that they were using truffles. And so one of the things they had on their menu was truffle soup. And so I told my wife we need to come and eat here, and she agreed because we both like truffles. And so they taste very earthy, but they're really it has a very distinct taste. And so I was a little bit disappointed, Brittany, because we went to have truffle soup, and so they had this soup, and then they came over with their little truffle, and then they kind of like gave me a couple of very thin slices of truffle on top of the soup, and that was it. And so it was disappointing, and that cost like $40, right? So yeah, and usually in Vegas you can get pretty good deals on meals, right? They're really inexpensive because they want you to gamble, and so they're willing to give up the cost of the meal and sell it to you at even sometimes below their cost, so that you're happy and you don't want to gamble, right? Yeah. So they're all over the place. But, again, the macroscopic, those larger organisms aren't going to be not going to be very clinically relevant. So we're going to really focus in on the yeast and on the molds. And so we're going to start talking about them now from a truly clinical medical perspective. And so we come back to the yeast, where I've just given an introduction of what they are. Now we're going to talk about them. We're going to talk about them a little bit more from a clinical perspective. Yes, Devin.
That is not true. There's a whole book called The Fungal Pharmacy that talks about different mushrooms, their medical uses. That's backed by like study after study after study after study. Like Reishi has 7
But what,
pages
what,
of studies that are
but
done
what
on
is
it.
not, but what is not true that I said?
That they're not clinically relevant.
Oh, okay. I meant from a infection perspective.
Right. There's even some mushroom fruiting bodies that they all have antibacterial, antiviral,
But
antifungals.
listen. I'm not saying from an antimicrobial process. I'm talking about from an infection process. They don't cause infections, Devin.
Oh, I see what you're saying. They're not clinically relevant because they don't do those things to us, not what they
That's
can
right.
do for us. I see. Sorry about
Correct.
that.
Right. That's okay. Okay. All right. So let's look now at because that's what this course is, Devin. It's not it's not a pharmacokinetics or a pharmacodynamics course with microbiology. It is a microbiology course for health sciences. So we're looking for how are these things going to cause problems, and that's not how they're going to save us. Now there is some of the saving thing, right? But we're looking mostly at infections. Okay. So if we think of, yes, Calvin.
Sorry, but don't molds like black mold from inside of a shower cause respiratory issues?
Um, Only a couple of them, Stachybotrys and Thotosporium, and but you have to be sensitive to them.
Okay,
Okay.
thank you.
Yeah. So I think I probably own just about every book that Devin is talking about. Um, so if you ever see me on campus and you want to read some of my reference books, you're going to be happy. I'll happily let you read them. But I don't loan my books out anymore because I don't get them back, and then I'm sad. So you can come and you can come and check them out. Okay. And I've lost many a good book. Right. Anyway, um, so if we think about the yeast themselves, remember they reproduce by budding. Some of them produce pseudohyphy, right? And so this is pseudohyphy. And one of the organisms that produces pseudohyphy is Candida albicans. So if we were to take a look at them, we could see these pseudohyphae growing. Now, the way that we classify yeast, or the way we identify them, is through morphology, right? But mostly it's through physical features like for instance, the biochemical test we're doing for the bacteria right now, right? And so here you can see there's different ways we can identify the yeast from the physiological characteristics that they have for what they use as a nutrient source and what they make as an end product of metabolism, right? So if we think about them, then this is Candida albicans, and this is what it looks like when you do a gram stain of it, right? So you can see the epithelial. So this was from a vaginal scraping. So here you can see the epithelial cells back here. Here's the nucleus of an epithelial cell, right? And then you can see all the bacteria back here, right? And they're gram negative. But then you can see how the pseudohyphae elements have been stained, right? And so here you can see these are pseudohyphae right here and right here. And then these are budding yeast. So if you see pseudohyphae and budding yeast and it's causing a problem in the human body, it's almost always going to be a Candida species. Now, Candida albicans is probably the most common species that causes infections in humans and other animals, but there's others, right? There's Candida glabrata, there's a bunch of others, right? But here you can see this is oral thrush, right? And so this is this is Candida albicans growing on the soft palate of the mouth itself and on the sides of the cheeks, on the insides of the mouth. Right, this is oral thrush, typically seen in kiddos who we haven't taught how to have good hygiene, or if they're for some reason their normal inhabitants of their mouth has been disrupted. And then this is looking inside the vagina, and so you can see there's a cervix here, but you can see then the yeast growing inside of the vagina. And the yeast should not show up like this. This is an indication. This is an indication of having a yeast infection. Mm-hmm. So yeah, and the different types of ways to get albuterol and like nebulizers and things like that. If you don't keep them clean, then they can be a vehicle for how these organisms get into our body. Mm-hmm. Yeah. So Caitlin, when I was second master's degree, I was probably 29. I was working for J&J, and I was pretty stressed out. I was taking 2 classes in the evening to finish my second master's degree. I was working 80 hours a week, and I was teaching 2 classes at ACC. I was pretty busy and pretty stressed out. And I woke up one morning. I had a kind of a sore throat, and I looked back there, and I had this growing in me. I remember it a lot whiter, right? But this is a little bit more yellow. So I have a microscope here at the house, and so I looked at that and I told my wife, I said, I have thrush. I need to go and see Dr. Chang. So in that day, my physician was Spencer Chen. He's really good, but he's in Round Rock. He's too far away from me now, so I had to change physicians. But Spencer Chen was really good, and we had a good relationship. And so every time I would come in, he would always say, well, what is my favorite microbiologist here for today? And so when I showed up this one time, I said, well, I came in for an antifungal because I have thrush. I have oral thrush. He said, no, you don't. I said, yeah, doc, I do. He said, no, you don't. I said, yeah, I do. He said, how do you know? I said, because I've taken a little sample from it and I've done my own diagnostic work. Here's the slide that I made. You can check it out. And he said, okay. So he gave it to his technician. She went to put it on the microscope or whatever. But he looked back there and he said, you do have oral thrush. He goes, yeah. He said, so what do you think we should do? I said, how about a 3-day prescription for diflucan? He said, oh, you know your drugs? I said, well, because I make the drug. This is a J&J drug. I said, so he said, well, that's a good one. So yeah, we'll go ahead and do that. He goes, now I have to ask you some questions. I said, here, doc, I've not ever been with a man. I've never had sex with a man. I've never engaged in sex with a prostitute. And so I went on and gave all these different things. He said, okay, good. He goes, but is it okay if we do an HIV test anyway? I said, yeah, that'd be fine. And so he was worried, that I had HIV because I was a young male who should not have had oral thrush, right? But really, when you're stressed out, the 3 systems of the body that can really can acquire an infection are the respiratory tract, the GI tract, are the 2 most important ones, right? And that's because the stress of the body make your immune system a little bit weaker, and then you're likely to pick up one of these opportunistic infections, like oral thrush or vaginitis, or what we call a yeast infection. Now, we're also seeing it in the immunocompromised showing up as anal thrush also, right? That's correct, Brittany. Yeah, because the yeast are everywhere, right? And so the babies, as they're suckling on the breast, then they can leave the yeast behind, and then it can exacerbate, and then babies can do that. But mostly it's because we're not keeping the baby's mouths as clean as we should be. Okay. All right. Yeah, it can be transmitted that way. Here's another important yeast, and this one really knocks our AIDS patients for a loop, and this is called Cryptococcus Neoformans. So Cryptococcus, you can see it's hugely encapsulated. Look at that. So here is the capsular network of the yeast itself. Right? So it's protected from phagocytes, right? So this one can get into the body, and if you're immunocompromised, there's not really a lot we can try to treat you with antifungals, right? But your immune system's not going to try to, and so if it gets into the brain or the meninges or the cerebrospinal fluid, it's just really, really bad. So years ago, I don't know, 8 years ago maybe, in Oregon, there was an outbreak of Cryptococcus, and I can't remember the species name. Hey, Calvin, if you look it up, Google Cryptococcus in Oregon cause infection. I don't remember. That's just it. So, but I mean, I think if you Google that, you'll be able to find out the species name. But anyway, people who were hiking in the forest were coming up with this. No, it wasn't that long ago. So people were hiking in the forest in Oregon, and they were picking up this really weird infection of a Cryptococcus species, and we had never seen that before. And so it made all the news for all the nerd publications that I get on infection. And so it was a really interesting thing. And it's just not like me to forget something of that type of magnitude, but evidently I'm getting older, and I think it's right. So it was Cryptococcus gotti. Thank you, Mr. Yeah. So Cryptococcus gotti. And it was causing infection. So you guys can look that up if you want. But it was really, and I don't remember what the outcome was or why we think that people were acquiring infections like that. But it was true that there were a lot of people acquiring infections from just hiking in the woods, which is something I like to do all the time. I have my own woods, but I love to go and just get lost in the wilderness. Yeah, it's an important thing to think about how that is, right? Okay. All right. Questions. All right. So if we look at molds, so those are some infections that were caused by yeast. Can't cover everything up because there are references and references of yeast. Did I ever show you my reference on the yeast? On the yeast? Did I ever show you my reference? Anybody ever see it before? Did I show it to you? I did. Yeah. So it's huge, but there's lots of references, and I have even more references on molds because I'm really interested in them. But this is Altenaria, right? I told you Altenaria is the principal infectious agent that really causes allergies. It's not really an infection, it's allergies, but it's an infectious agent, right? So we have Mycelia, right? That's either aseptate or cenocytic, or it's septate, right? So we're looking at molds now. And then the spore formation can either be asexual, or the mold can have asexual and sexual reproduction. Okay, so this is the mycelia and the hyphae, right? So the entire structure, Julia, is the mycelia, and then one unit in that mycelia is a hyphal structure. Okay, so if we think about that, then really the molds are defined as multicellular spore-bearing achlorophyllous organisms which multiply sexually or asexually and whose usually filamentous somatic structures can be septate or aseptate. And then a caveat that I like to add to the end of that definition is the nutritional needs of the fungus, of the mold, are facilitated by the enzymes cellulase and or chitinase. And so depending on what type of mold we're looking at, this are going to be the parameters that will begin the basis for its definition. It's not the perfect definition because every organism is different, right? But if you see this is an orange, and the orange, of course, has got mold growing out. And if we take a micrograph of it, this is what the mycelia will look like. And you can see the septa in the actual structures themselves. And so this, if you're looking at this, are septate versus aseptate mycelia. And if you look at them a little bit closer, you can see the cross walls and the septate structures, and no cross walls in the aseptate structures. Okay. So if we think about them, the mycelia, whether they're septate or aseptate, will propagate, and they propagate apically, right? What does that mean, Devin? They propagate apically. What does that mean? Apically.
It's escaping me right now.
Okay, that's okay. Calvin, what does apically mean? Naeli? Apically means it grows at the
It's
very
returning to the apex or point.
That's correct. It's growing at the very top, right? So the growth is going to continue like this, much like a plant would, right? And so, yeah, they have some characteristics that are like plants. They produce spores. The spores get liberated into the environment. The spores get either picked up in our clothes or on animal fur or by the wind or whatever, and they go, and then they simply go and get distributed somewhere. And if nutrients are available, those spores germinate, and you have a mycelia start to grow out of the spore, and eventually that mycelia will form a new colony. So the colonies of mold, so a colony of a yeast cell will grow in about a day. The colony of a mold will grow and become mature in about 3 to 5 days. But the colonies of mushrooms on the average are going to take longer, right? Might take a week, might take up to 4 weeks for the colonies to become mature. And so when we grow mushrooms, and Devin can attest to this, Devin, tell us again what species of mushrooms you guys are growing.
So we grow lion's mane, Whitehairia, reishi, Piopino, chestnut. We grow a cousin of lion's mane called comb's tooth. We grow 3 different types of oyster. We grow, what else do we grow? Oh, and shiitake, and maitake, and nameko, and enoki.
Yeah. So Julia's got a question for you, Devin.
No, I work at a mushroom farm. So we grow mushrooms
I wish she was
for
growing mushrooms for this class. That would be
for
really
food
great.
and medicine. I could. We need to talk after class. To talk to my boss.
Yeah. Yeah. So I mean, it is. I mean, I don't know, Devin. Could you maybe, if I can get it okay with ACC, do you think maybe you could give a little tour?
We're in the process of moving right now, but if you can
Okay.
get
Okay.
it,
Okay.
if you can get it cleared with ACC, this would be something we're moving like within the next month or so, maybe 2. But if
Yeah.
one, it could be cleared for whenever we move and get set up there. It'll
Well,
be a lot.
check with your bosses, because I think it would be just fabulous. So when I was in Magnolia, Texas, I went oh, one of my really close friends. I was in Magnolia, Texas, and they invited me to the what is it, Renaissance Festival out there? Is that what that was?
Yeah, out in Plantersville. Out in
Okay.
Plantersville, yeah.
So, but for somehow or another, I found this mushroom farm out there, right? And so I was supposed to meet my friends at the Renaissance Festival, and I was 4 hours late, mostly because I started talking to the guy who owned the mushroom farm, and he invited me in, and I just got to walk around his farm, and I was just fascinated about everything. That was a really cool thing, right?
Yeah, they have one in Madisonville, which is like kind of, it's near Plantersville, but they have a huge mushroom farm in this tiny little podunk town in Texas because of
Yeah,
it.
I don't remember where it was, and I don't even remember the name of the company anymore. But the guy was really nice, and he said, "Well, you want to come on in and look at it?" Oh yeah. And so yeah, I was much more interested in the mycology than I was in the Renaissance Festival. Was fun, but I had a lot more fun at the mushroom farm. Oh, I'm sorry, Pedro. He'll get to look at this. All right, I'll ask Devin. But hey, Devin, I didn't mean to put you on the spot or put you under pressure. Uh, If you can't do it, that's okay. I just
I
think it'd be
don't
really cool.
I don't feel any pressure, but yeah, we
Okay.
can talk about it afterwards.
Okay, so if we look at asexual spores, the spores of the molds that produce the hyphae that are aseptate, the spores that are produced by them are called sporangiospores. And the spores that are produced by those organisms that are more evolved, the ones that have septate mycelia, are called cnidiospores. Okay, I'm not going to talk about every single organism, I can't, but I'm going to talk about some of the ones that are most likely to cause infection. Here are the way that the spores can be liberated or can be grown, right? So this are conidiospores. They're growing on a conidium, right? This is a conidium, and you can see the spores are just available to the environment. So if a nice breeze comes in, these things break off and they get carried away. Here, a sporangiospore is formed inside a vesicle, right? And so if you look at, here's the vesicle itself. And so therefore you have the spores inside, and eventually this vesicle breaks, right? And the sporangiospores get liberated into the environment. And so there are two different mechanisms by which spores are propagated asexually. These are asexual spores. They get propagated and then they get released into the environment. Okay. So there's lots of difference, right? But almost all sporangiospores are inside a vesicle of some sort. And the conidiospores, there's lots of different ways that they get liberated, but they're never in a vesicle. They get liberated freely into the environment from the actual vegetative structures of the fungus itself, of the mold. Okay. So again, just to recap, for yeast, we use budding and pseudohyphy, and then the biochemical procedures that they have in order um, to identify them. With the mold, it's the mycelia and it's the spores. And then with the macroscopic, it's also the mycelia. They also have clamp connections, right? So I don't know if you know this, Devin, but clamp connections are found in the Presidio Mycoda. And there are these little junctions that form between the two different hyphal elements, and then it can be exchange of information between the two different cells. That's right. That's right. Okay. So we're going to look at some of the organisms that can cause infection. We're going to look at the Zygomatic and the Ascomycota. And then we're going to look at the Presidio Mycoda, which the Presidio Mycoda are the larger organisms, which we're not going to talk about too much because they don't really cause infection. So again, if you look at molds, right, they can be parasitic or saprophytic. I'm glad you know what those terms are already. They form filamentous structures known as mycelia, and each of the mycelia are composed of single cellular units called hyphae, right? Their cell walls are composed mostly of cellulose, but they have chitin. They're slower to propagate when you compare them to bacteria, but faster if you compare them to a human. They're all aerobic and they like pHs that are acidic. Yes, sir, Calvin.
Would it only be a septate hypha that grow apically?
Nope. Nope. All hyphae, all mycelia grow apically.
Thank you.
You're welcome. Okay, so if we, I'm sorry, Nayeli, I'm not being able to keep up with the chat. I'm not really good at multitasking, so but anyway, okay. So Calvin's keeping me honest. So if we think about this, then you can see that that you can look at, these are the aseptate organisms. This is a beautiful micrograph of the aseptate hyphae. You can see how there's no cross walls. And of course, what is this thing right here? What is this thing right here? That these
CFU.
Yeah, well, you're right. It is a CFU, but it's a spore. That's right, it's a spore. But you're right, Calvin. It is a CFU. It is a colony forming unit, right? Uh, look at Caitlin. She's being funny with this. It's a UFO. Yeah. So we're going to talk a few about them, right? So with the But these lower organisms, right, these less evolved organisms, they have aseptic mycelia. The old term that used to be used, they used to be called the phycomycetes. And if you run into an old physician, uh, they might use the term phycomyces, and then everybody just kind of looks at them funny because that term is not used very much except for by older physicians. They're saprophytic or parasitic. They're primitive. They live in soil and water. They are aseptate or cenocynic, and they can cause infections. But all molds, when they cause infections, they have to have oxygen, so they can only cause infections where there's oxygen available to them, right? So mostly we're going to see them causing infection in the lungs and on the skin. That doesn't mean they can't cause infections in the brain, um, but there has to be some special circumstances for why that might happen. So let's talk about the Zygomatic. They're called the Zygomatic, right? Again, myco meaning mycelia, but zygo meaning that at some point they form a zygote, right? And so here you can see um, the different mycelia coming together. Oh man, you can see the different mycelia coming together. So this would be mycelia number 1, and this would be mycelia number 2. We don't really call, I mean, some people do, but we don't really call them male and female. We sometimes say the plus or minus, but they come together and they form a zygote. And this is a sexual state, and inside of there are sexual spores. Okay, but for the most part, these things, when they form these zygotes, if you will, then inside of there are going to be genetically unique organisms, right? Of them, there are several different groups. The first group are called the Catridial Mycota. The chytrids are responsible for recycling nutrients. Up until about 20 years ago, we never saw them causing any infection in any animal. Um, we now know that they are causing the wipeout of lots of species of frogs. So something has happened to the environment that now the chytrids are becoming very pathogenic to the amphibians, right? And so they're wiping out whole species of frogs all over the world. And so it's really interesting to see that. And there are people studying that phenomenon right now about why the chytrids have become so pathogenic all of a sudden, right? But mostly, for us, right, um, they're mostly involved in the recycling of nutrients. However, I have made the observation that, I have that creek that's running in my backyard. I have made the observation that I have not seen or heard a frog in about 10 years. Right before that, they were everywhere, and now they're not. Now, from time to time, we see toads, and toads are all over the place, but I'm talking about a true frog. Yeah. So what that means to me is there's been some kind of change in the environment. Um, so I don't know what's going on. Have you guys seen a frog? When's the last time you saw a frog? Roly poly. Yeah, I used to see them all the time, right? Used to see the tadpoles all the time, right? But toads produce tadpoles also, so you have to be careful. Yeah, yeah, they used to be everywhere, not anymore. The Trichomycota are pathogenic fungi for insects, and so when you watch the Paul Stamets video, get on the lookout for the Trichomycota, right? He's going to talk about them because he's got patents on a lot of them. I have in my refrigerator right now, I have an organism of fungus that will kill fire ants. And I have to find out whether or not Stamets has the same organism, because if he doesn't have this organism, then I can uh, do some research in it and then publish a paper, and then I'll have a patent on it. But unlike Stamets, who I love, I think he's absolutely great. He sells his fungi. I would just give this to the world. I would just make it free for anybody who wants it, because I don't really like fire ants and they don't belong here. Okay. So yeah, so I'm not willing to give it to anybody just yet, because if it's not what I think it is, I don't want to release a pathogenic organism to insect species if it's going to kill all the ants. I just want to target fire ants. Okay, and then when we move on and we'll talk about the organisms that have septate mycelia, right? We're going to be looking at the ones that we're going to be really interested in. And I know it's time for lecture to be over. I'm going to go a little bit longer because the lab is really short today. So I'm going to go a little bit longer, finish this, and then we'll go to lab. And you'll still get out early from the class today. Okay. So if we look at the Presidio Mycoda, right? Remember, the Presidio Mycoda themselves, these are the fleshy organisms, right? This is the basidium right here. And you can see the Presidio spores that are produced on the basidium themselves, right? And so that's a really pretty cool thing. But these things are beautiful. And the variety of them throughout nature is just so beautiful. If you know what you're looking for, you can find them, and they're just so gorgeous, right? And they're so perfect for the environment because they're just recycling nutrients mostly. The Ascomycota are the group that we are interested in, right? Because they can produce infections with us. So if we think about them for a minute, the Ascomycota have the ability to reproduce sexually and asexually, and this is a sexual state right here of them, right? This is what we call an ascocarp. And inside of them are the ascospores, and those ascospores are the unique sexual structures, and therefore they're genetically important, right? So this, if you look at this, and here's some wicker, and all these little dots that look like pepper on them, those are all an ascocarp. So these little dots, they're all ascocarps, right? And so now that you know what an ascocarp looks like, today when you go for a walk or you're outside in the yard, pick up a twig, and you'll see these little black structures embedded in the twigs, and those are ascocarps. Those are the sexual cycles of the fungi, and they're everywhere, right? That you can't get away from. This was my wife. I don't know if I told you this. My wife and I used to own an antique business. We still have a lot of antiques, but we were in business till about 2000, and then we closed up the business because they had the big stock market crash, and a lot of people lost a lot of their um, retirement, and so they didn't have a lot of extra money. So the antiques weren't selling very well, and so we were just paying rent for this place that we're at for nothing. We weren't making any money, so we just closed it up. And most of it was a hobby for us. We all had full-time jobs. We just did this kind of on the weekends. Okay. But anyway, this was wicker furniture. I was in Westlake looking at antiques, and I was looking at this wicker furniture, and the lady kept me, "Oh, are you interested in my wicker?" I said, "I am." She said, "Would you like to buy it?" I said, "No." I said, "But I'm really interested in the fungi that are grown." And my wife looked at me and gave me the face, like, "You shouldn't have said that," because I was quickly asked to leave her property because she said it was dust. And I said, "No, ma'am, this is a fungus growing under wicker." So I got asked to leave the premises. She was nice about it, so I'm not too problematic. An anamorphic state for the Ascomycota is when the anamorph, write that down. An anamorphic state for the Ascomycota is when the fungus just reproduces asexually. A telomorphic state, telomorph, is when they can reproduce sexually and asexually. Okay? Be sure you know those terms. Right? So a telomorph. Let's talk about some of them. So there are 5 true fungal pathogens, true pathogens, right? A lot of them are opportunistic, but there's only 5 true, where there's hundreds of thousands of pathogens that are other organisms, right? Where the fungi, there's only 5 true pathogens. And these are pathogens because they are dimorphic, right? So what does that mean when you say they're dimorphic? What does that mean? They're dimorphic. What does that mean? Caitlin, what do you think?
They represent in two distinct forms.
Two distinct morphologies, right? And so that's really cool. At room temperature or in nature, they are molds, right? But when they get into the human body, they become yeast. Isn't that interesting? And so that's a huge advantage. It's a huge advantage for these organisms. So let's talk about them, and then we'll talk about why dimorphism is important. Histoplasma capsulatum, right? So this causes Darling's disease, right? And so this is what it looks like if you were to look at its spores. This is what it looks like when it's infecting the lungs. Now, if you see a radiograph of healthy lungs, the lungs are going to be really, really dark because when they're expanded and they're filled with oxygen, with air, then they look really, really dark. Okay. So here you can see there's some infiltration in the lungs here and here, but also here, right? And here. There's some infiltration in the lungs, and that means that there's typically something growing in the lungs themselves, right? So this particular organism is associated with the bird poo, right, and also bat guano. And so you can see epidemiologically where it's found, and it's found in Austin. We have a lot. We have a lot of bats here. They're not here right now. At least I don't think they are. It's too early in the spring. But the Mexican freetail will be coming back, and they have 3 colonies in around Austin. Right, there's one underneath the Congress Bridge, right by the newspaper. Right, there's one in off of the 620 overpass in Ramrock, and then there's one near the Pennybacker Bridge out in the northwest Austin. Right. Um, well, um, no, the our bats, Caitlin, most of our bats are migratory. So the bats, the Mexican frito, will migrate back to Mexico during our winter, and they'll come back in the spring, and they'll be here through early part of fall, and then they'll go back. Right? They are stinky, right? But the other thing to make note is that the the feces of the birds and the bats, the guano, that's where you're going to find this pathogen, right? It's going to be found associated with those particular substances, right? So I can remember as a little kid, I'd go to my grandmother's house and we'd clean out our chicken coop, and my brother and I would be flicking chicken poop at each other all the time. And both of us test positive for the antigens for histoplasma. Yes, Calvin.
So this is all part of Unit 3, correct?
Yep, it's all part of Unit 3.
Okay, thanks.
So, spelunkers. What's a spelunker?
Somebody who dives in a cave for...
Somebody who goes and studies caves, or just kind of likes to be in caves, right? So we do get to see this in some spelunkers because of the guano that's associated with. But really, if you're immunocompromised and you're just hanging out by a bat colony, you're walking underneath the Congress Bridge, you could pick this organism up. And some people do. And if you have a really good, healthy immune system, you're probably going to be okay. But if you are immunocompromised, it could kill you, right? This is another one. This is Sporothrix schenki, right? So this is another dimorphic fungus. This one typically infects skin, and it's sometimes called rose handler's disease. And this is what the spores look like. They form this little collarette. But you can see how this particular person got an infection, right? And they were handling roses. They got stuck by the thorns. They can be found on the pyracantha too. Anything that's got thorns on it, right? And then you can see how the infection started to spread, right? My friend, microbiologist, was also an award-winning rose producer, And so she had these beautiful roses growing, but she would always walk around in her garden barefoot. And I always told her, I'd tell her, hey, Shelly, you need to wear some shoes when you're walking around those roses. That's okay. But, she got stuck by a thorn. She cleaned it with iodine, thought it'd be okay. Um, and then she went to bed, and about 4 o'clock in the morning, she woke up with what she said was the most intense pain she had ever had. And what she had was a fulminant infection of Spore Trichomycota. And so they rushed her partner, rushed her to um, the hospital, and they debrided her entire leg because the fungus had actually spread up her leg already. And so she stayed in the hospital for about 4 days. They got the fungal infection under control, and she's okay, but the scars are still there. It's an amazing how quickly these things can move. This is the most important of the pathogens. This is Coccidioides immitis. Hey, have you seen those commercials on television for Imbro or Humira? It says, tell your physician if you've been in parts of the United States where certain fungal pathogens are found. Have you seen that? So this is the one they're talking about. And so you can see that epidemiologically, it's found all the way up through Texas, all the way up through some of the southern parts of California. And so you can see that in Arizona, New Mexico, it's there. And so in some places driving there, you can see these signs that basically will say, please keep your windows up because of fungal pathogens found in the soil, right? And so they get blown around and you can breathe them in, and then you can come down with an infection. Again, they need a lot of oxygen because they're aerobic. And now you know, Naeli. But you can see how they can cause infections of the skin or of the lungs, right? One of my closest friends that I worked with at AOK Obeyo, she grew up in Phoenix, and she told me the story because we were working with coccidioides in the lab, and I said, I'm gonna have to get a positive control for coccidioides. And she said, you can just use my blood. I'm like, what? She said, yeah, I test positive for it because I've been infected with it. I'm like, oh my God. But I couldn't use it because it wasn't a qualified control. But I did take a little bit of her blood, and I did test her blood, and she surely did test positive. So she told me the story, but her father tells the story a lot better. And the young lady told me, I knew I was really sick because my father never really slept with me, and he wouldn't leave my side for about 2 months. So they were digging a swimming pool in her backyard, and she breathed in some of the spores of coccidioides, and she came down with a fulminant infection that almost killed her, right? So this thing's really, really, it causes valley fever. It is really, and look where it's located, right? Throughout Texas, all the way up and through California. Okay. And then the last organism is Blastomyces dermatitis, right? And it is found associated with bodies of water. So you'll find it, we typically, when we talk about them, we look at the Mississippi, Ohio river valley basins, but it's also found, around Town Lake and around Lake Travis and all these other places, right? But you just have to be careful, right? If you're immunocompromised, you just have to be careful about these infectious agents because they can cause problems. Okay. So if we think about this, you can see here again, when you take when radiograph is taken of you, you always want to take a ventral and a lateral view, because if you look at the ventral view, right, you can see that maybe there's a little bit of infiltration, but if you look at the lateral view, look at the infiltration of the fungus all throughout. If I was in a lab and a physician called me in to take a look at an infection, I would think that this is Staph aureus or a gram-negative organism. We would debride that. I'd take samples of that. I would go and investigate it. And real quickly, what I'm going to find are yeast, because the organism the 4 organisms we just talked about, the true fungal pathogens, Histoplasma Capsulatum, Spore Trichomycota, Coccidioides immitis, and Blastomyces Dermatitis, they're all dimorphic. And so that's what makes them really true pathogens, right? So dimorphism, if we look at that, dimorphism allows for the organism to have an advantage because they can grow more rapidly, right? Only takes them about 45 minutes to become 2 cells, whereas a mold, it would take them 3 to 5 days to develop a colony. They can fend off the immune system because as yeast, they can form capsules. And because they're smaller, they can obtain nutrients a lot quicker. So this is a huge advantage for this infectious agent. It's a disadvantage for us. Okay. Again, pathogenic, they're dimorphic. So in nature or at room temperature, they're molds. But once they get in the body, they will become yeast. Okay. And that's a huge advantage. All right, a couple more. So we're going to look at some of the opportunistic infections. And when we look at opportunistic infections, we're mostly talking about ringworm, right? And so if we think about ringworm, right, it is a fungal infection. And it presents itself lots of different ways, right? So here is hyperkeratosis of the toenails, right? Yes, Calvin.
So if there's a fungus presenting in one of the big toes, could that also be a ringworm related thing in the back
Yep.
of the leg somewhere? Is that like,
Yep.
does it, okay, so it
Yep.
does translocate like that.
It can. And as a matter of fact, ringworm is one of the more contagious organisms that can translocate. Yeah. You can have it on your hand, you can have it on your feet, you can have it on your face, and you can have it in your groin, but it might have just started on your hand, right? And it can translocate like that. The difference between having hyperkeratosis of the toenails, so if mostly older people who are missing minerals and vitamins, is that they're really hard to cut. Impetigo is a bacterial infection, Caitlin. We'll talk about it. It's group A strep. But if we think about that, then if it's a true fungal infection, you can just pick at the nails, and if you pick at the nails just a little bit, they're going to flake off, and that's an indication that you have a fungal infection. And if you have these in your toenails, probably your fingernails are the same way. There are 3 different etiologies, so 3 different organisms that cause ringworm, and that's trichophyton, epidermophyton, and microsporum. 3 different species, 3 different genera that have several different species. If you see SPP, that means that there's several species of these organisms. So if we think about this for a minute, right, these are all going to be the etiology of ringworm. Now, when we talk about ringworm, right, this slide is hard to see, but the medical institution does not like to say you've got an infection by trichophyton. What they like to do is they've changed it, and they basically have said you have an infection of tinea, right? And so the dermatologists who came up with this idea said, okay, good, we have a genus, we need to have a species name. So what they decided to do is put the part of the body in Latin as a species name where the infection is occurring. So for instance, right, Tinea pedis would mean you have ringworm of the foot. Are you with me? So now we have Tinea capitis, ringworm of the head, Tinea pubis of the genital area, Tinea pedis of the feet, Tinea corpus of the body. You have Tinea ungum of the nails, and Tinea manum of the hand, right? But there's others, right? Tinea barbare, there's of the beard. You could go on and on and on, right? All you do is you just said tinea, and then you add part of the body where ringworm is causing the infection. Remember, this is a fungal infection. The center of that infection is right there, and then it's growing radially, so you can see the outer parts of the ringworm itself. Yes, Calvin.
If the patient is presenting as having a toe that's very fungusy, and then they have a spot on their back of their leg that looks like a ringworm thing, is that always ringworm, or can that be presented as?
It can be presented as something different, so you should. A good dermatologist will test both sites to make sure it's the same organism. But
Okay,
empirical treatment of the skin can happen really quickly. Of the toenails, it's much more hard to get rid of, and so a lot of times you have to use long-term antifungals in order to get rid of it. Okay, sometimes you will remove the nail completely and allow the nail to grow back again if it's really serious, right? Yep. All right. Malassezia furfur. It's not really ringworm, but it's in the same grouping just because we don't have any place else to put it. But Malassezia furfur causes Tinea Versacolor. This is what we call sunspots. How many of y'all knew that sunspots are produced by ringworm? Right, so we typically see these in our darker complected our colleagues or citizens, and it usually happens in the winter, right? And so you start to see these spots. That's different than if you got sunburn and then you start to have your skin flake off, right? Slough off. That's a little bit different. This happens so that you have this mottled looking skin, and it happens when you haven't really been in the sun, right? And so everybody can get this type of problem, right? But it's really highlighted in our darker complected individuals. Malassezia furfur is the organism. And as we begin to close this lecture, I think Calvin brought it up. So sick building syndrome is a real thing, right? The two organisms that are usually associated with sick building syndrome are Stachybotrys and Cladosporium. And that's because they produce mycotoxins. Right? The mycotoxins themselves can affect people differently. Sometimes they cause them to have respiratory issues. Sometimes they cause them to not be able to function properly. Sometimes it causes a little bit of pain. Ketomium and Mucour, those are really different organisms. So Ketomium is an Ascomycota, and it's really good at breaking down like fabric. And Mucour is another organism that's a fast-growing organism, but it's not usually associated. Those are not usually associated with sick building syndrome. Okay, but if we think about that, then here is a house that was after a hurricane, and so you can see that the water got this high, and so they took the water out. They dried the place up, but then they did the worst thing. This was on a Florida house after a big hurricane. They did the worst thing is they basically locked the house up and didn't allow for any kind of ventilation. And because it was wet, the mold went crazy. Right. This type of house sometimes will get destroyed, but it could be saved. All you do is remove everything from the internal parts of the house, disinfect, and then put in new sheetrock, and then the house would be okay. But there are less than 0.01% of the population that are hugely sensitive to the mycotoxins. I am not one of those people. I grew up in Corpus Christi, and I can tell you in Corpus Christi, the two things you had, and if you were rich or poor, it didn't make a difference. You had roaches and you had mold in Corpus Christi, Texas, right? So I never had a problem with mold. But when I moved and went to college, one of my friends got a real good deal on a little place, but she was sick all the time, and that's because of the mycotoxins that were in the house. And so I then went in there and I took some agar plates from the lab. I taught labs at Texas State. And I grew Stachybotrys, and so we were able to show the landlord that the little house had. I didn't call it sick building syndrome. I said that there was mold that was making this person sick, and she would like her deposit back and her money she paid for rent because she needs to move. And so she came and lived with me for about 2 months, and then she found her own place. But it was a pretty serious problem. But remember, most people are not going to have a problem with sick building syndrome. Where would you find it? You're going to find these organisms anywhere where there's water, right? Around washing machines, around HVAC systems, things like that. My friend in Corpus called me up one day and said, "Hey man, there's a house on Louisiana Street that they're selling for $100,000." I said, "What's wrong with it?" He said, "They say it has mold in it." I'm like, "Okay." Well, my mom was still alive, and so I went and I told her, "Well, I'm going to be down there this weekend. We can go check out the house, right?" So I went to make sure my mom was okay, and then my friend and we went to see this house, right? And when we walked in, I said, "This is a beautiful house." I said, "Where's the mold?" And she showed me, and so it was a patch about this big, right underneath they had in Corpus, the older houses didn't have central air. So what they do is they cut a section out of the wall and they put a window unit in there, and then and then they had air conditioning. Well, the air conditioning had leaked and it caused the mold to grow. And I said, "This is it?" He said, "Yeah." I said, "Dude, you need to buy this house." And so he bought the house. We fixed it, right? I disinfected it for him. We put in new sheetrock. And now he's got this beautiful home that's completely paid off because he got a really good deal on it. And Louisiana Street, if you've ever been to Corpus, it's kind of in the older uh, district of Corpus Christi right now, right near Swan Hospital. And these are beautiful homes that are much more elegant. They're really beautiful. So you got a really good deal on a home. I tried to do the same thing with a house in Dripping Springs. There's this really big, beautiful home that the contractors didn't do a really good job on, and they let the insulation got wet, and then mold started growing. So they lost the project, and they gave it up back to the bank, and I offered the bank $100,000 for the property because I was going to fix it up, and then but they wouldn't do it. So they tore down the house, and then they separated the acreage into 5 lots, and they built 5 houses on it, and they made a lot of money. Right? Well, you can't blame them for that. But in the 1990s, you could take a class over the weekend and become a mold remediator, and you could go into places. Some people went in like this, and that would sick out of people. And then they would basically say, we can clean all this up. We can make your house safe again if you'll move out for a week and let us do our work. And so the people would move out. These bunny suits would come off, and the radio would start playing, and they would take a 10% bleach solution and they would wipe down everything. Some cases they had to replace the sheetrock, but really it's really easy to fix, and they made a whole bunch of money. So now if you own a home, on the back page of your insurance, there's a rider that basically says, "We do not cover anything associated with windstorms or with mold." So they are not going to cover anything, any damage to your homes that are done by hurricanes. You have to have special hurricane insurance if you live in a hurricane zone. And then they don't cover anything with moles, and you can't buy anything. Nowadays, if you live near a creek like we do, we have to have flood insurance in order to protect our property, because the banks no longer and the insurance companies will no longer cover if your house gets flooded, if it's in a floodplain, unless you have special insurance. Yes, sir, Calvin.
I was curious if, for the safety of the workers, if there's some sort of treat that they can do prior to the people going in there to clean to nullify the mold, like introducing a very high heat or using a UV light or something that
Nope.
would basically kill the mold before they work.
Nope. UV is ineffective because of the mycelia, the way it grows. So really the only way to do it is to hit it with some type of chemical. That's the best way.
Okay, so they just basically go in there with protective stuff and then scrub it off, and that's the best.
So this Calvin, the point I was trying to make is this protective stuff was mostly for show, because a lot of times they would tell
Right.
they would tell the people in the house, "We'll fix this problem for $10,000. That's way overpriced." That's why the insurance started to not cover mold, is because it was really more fraudulent than anything else. Yep.
Okay, so those suits are kind of like the Monsters Inc. They go in there and they're like removing the sock, but it's just a sock and a Monsters Inc.
Correct. Okay.
So
All
it's the
It's a what? Oh.
It would be theater then, like it's
Yeah,
just
it's
kind of
the
a
it was
production.
theater, right? So these guys who were mole remediators, and I remember seeing them. There were a lot of them. They would take this course. They would have a certificate saying I am a mole remediator. So they were a grocery store worker during the week, and on the weekend they had this little magnetic sign they would stick on their car or their truck that basically says we remediate mold, right? And so therefore they did that job over the weekend, and a lot of times it was very lucrative until the insurance industry caught on and basically said no more, right? I leave you with a couple of slides. Um, no, not really. Um, the thing that's really profitable right now is if you can remediate asbestos, because there's a lot of older homes that have asbestos. Now you're playing with fire with that. I do not recommend, if you don't know what to do not try to remediate asbestos because it's bad. Okay. Anybody who's got popcorn ceiling, and if your house was built in the 70s, you probably have asbestos in your house because most of that popcorn ceiling was asbestos. Okay. All right. So I leave you with a couple of things. Number one, the symbiotic relationship between a fungus and algae. And so you can see here, these are the different types of lichens, right? And they are a symbiotic relationship between the fungi and an algae. So about 3 different species of fungi, 1 species of algae. And so this is just showing um, the versatility of the molds and how they can form relationships with different organisms. So if we think about it as we close here, the fungi are my favorite kingdom, right? Yeah, they're involved in infections. They do produce allergies. They produce poisons. Agricultural damage, right? In the year 2000, there was very little wine coming out of Napa because of they had grape blight there, and it just destroyed a lot of their grapes, right? But the benefits of the fungi far outweigh anything they do that's bad, right? They decompose organic matter, right? They form association with the root tips with plants. They help to mitigate the nutrients between different plant species. They produce antibiotics, alcohols, organic acids, vitamins, food, and they are also a lot of the cheeses that we have. They're flavored because of the end product of metabolisms of the fungi. So, ladies and gentlemen, to me, even though many people fear them, I think that the fungi are kick-ass. They are the most beautiful and my most favorite of all kingdoms. May I take your questions? Anybody have any questions? Okay. So I kept you a little bit over, but I told you the lab is real short. So why don't we take 30 minutes? Why don't we, no, let's take 12 minutes. Let's come back at 9:30. No, it's 9:23. Let's come back at 9:40. We'll have lab. It's real short. And then we'll talk a little bit, and then you guys are going to be free for the weekend. Okay? I'll see you guys. I'll see you guys in just a little bit. Let's target 9:40. Bye bye.